Good morning, everybody. As an example of research in viticulture, I will show you the experiment of my PhD project that was on the effects of biostimulants on secondary metabolites in Vitis vinifera. When soil water stress combines with high atmospheric water demand, the water that flow into grapevine leaves often is insufficient to compensate water losses through transpiration, resulting in a depression of leaf water potential and consequently in a reduction of leaf gas exchange, namely stomatal conductance and photosynthesis. Soil water stress combines and interacting with heat and high radiation level has also a lot of effects on the biosynthesis of primary and secondary metabolites, especially in the berry. And in particular, this extreme condition may promote the accumulation of sugar and inhibit the formation of secondary metabolites, like, for example, anthocyanins and flavonols, with a possible imbalance in grape quality with an elevation of the alcoholic content and changes in polyphenolic composition. Extreme condition may also lead to a phenological advancement that may deepen the negative consequence of climate change because clusters are exposed to heat that occur earlier in the season. Polyphenols are product or byproduct of the phenylpropanoid pathway and are very important both in plant cells and in grapevine in the red wine because in plant cells have an antioxidant function by preventing the formation of reactive oxygen species and by the direct scavenging of these species. Protecting the photosynthetic process. In red wine, mainly anthocyanins and flavonols are responsible of flavor, color, and stability against oxidation processes. In the last few years, scientists have studied the role of biostimulant as possible promoter of short-term plant acclimation against the environmental constraints. A biostimulant is any substance or microorganisms applied to plants with the aim to enhance nutrition efficiency, abiotic stress tolerance, and crop quality traits and plant performances in general. There are many categories, and among them, seaweed extract that were used since antiquity as amendments to increase crop production and soil fertility. In particular, Ascophyllum nodosum is a macroalgae and seems to modulate the activity of antioxidant enzymes, increasing the biosynthesis of antioxidant compounds, like, for example, polyphenols, and thus eliciting plant physiological and biochemical responses to the abiotic stress and enhancing plant performance in general. The aim of our experiment was to investigate the physiological performances of Vitis vinifera and the biochemical adjustment induced in grape berry skin by an Ascophyllum nodosum extract foliar application in field condition. As I mentioned before, the Ascophyllum nodosum is a brown macroalgae rich in growth stimulating compound, like, for example, carbohydrates and amino acids. Here you can see the table of the main chemical characteristics of the extract that we used, and as you can see, this extract is very rich in amino acid. Among them, for example, phenylalanine is a precursor in the phenylpropanoid pathway that I showed you before. Other amino acids, like, for example, proline, act as osmolytes, helping plants survive osmotic stress. And we know also that amino acids are regulator of the ion transport and act as antioxidant compound because also them reduce the oxidative damage due to raw species. The experiment was carried out in 2 seasons, 2016 and 2017, in field condition. We had 2 treatments, SWE, where plants were treated with the Ascophyllum nodosum extract, and control, that where plants were treated only with water. And the 2 treatments were performed 1 week after full veraison and 50 days after, just to match the main stages of accumulation and biosynthesis of sugar and anthocyanins in the berry. In field, we have some tools to monitor ecophysiological parameters, like, for example, we have this infrared gas analyzer, CIRAS, that monitored the photosynthesis and stomatal conductance, so leaf gas exchanges. We can use pressure chamber to monitor the midday stem water potential, and we also used a fluorometer to measure the chlorophyll fluorescence, so in general the efficiency of photosystem 2. Measurements were taken on 10 homogeneous leaves, from 10 leaves from 10 homogeneous plants per treatment at 3 stages: full veraison, that was pre-treatment, mid-maturation after the first treatment, and full maturation after the 2 treatments. We also collected lots of berries to perform some analysis on soluble solid concentration, titratable acidity, pH, and also on the berry weight. At the same 3 stages, we collected also lots of berries to make anthocyanins, flavonol, and hydroxycinemic analysis with HPLC. Some results. These tables show the ecophysiological traits. For here you have the 3 stages in the 2 year, the 2 treatment, and these are the parameters: photosynthesis, stomatal conductance, the quantum yield efficiency of photosystem 2, and the stem water potential. As you can see, here in red you have the statistical significativity, and as you can see, in SWE we had a higher stomatal conductance than in control, often, sometimes, and also a higher photosynthesis than control, also under severe climatic condition that we had in mid-maturation 2017. That was a very harsh season. Here I want to point out this control value that is very, very low, under the threshold of 50 millimole, that is the threshold of severe water stress in grapevine, instead of a good value of SWE. And here you have a lower PN for control, but also a lower quantum yield efficiency for the control, that witness signs of metabolic limitation to photosynthesis. As regards the berry composition and productivity parameters, we had only differences in total sugar, with a lower total sugar content in SWE than in control, in both seasons. And this is really important, is a good results for the viticulture of nowadays. And we had no effect on berry weight, but I want to, so this reduction in sugar content is not due to a dilution, because the two treatments had the same berry weight. And also I want to point out then that in 2017, that as I mentioned before, was a very harsh season. We had a berry weight that is almost the half of the year before. Here you have a series of graphs. Don't be scared about the number of graphs, because they show all the same things. So the anthocyanin content in the berry skin with the HPLC DAD. And here you have the 5 main components of this red local variety, that is Sangiovese. And here you have the total anthocyanin content. With the 3 color, you have the 3 different timing of sample. The 2 here, the 2 treatment in the 2 here, and the letter is the significativity. As you can see here, in 2017, we had a higher amount of total anthocyanins, maybe due to the harsh climatic season and or caused by a berry dehydration with a passive accumulation of anthocyanins in the berry. I want also to point out that in both here, SWE at full maturation had a higher amount of anthocyanins, in particular of those anthocyanins that are cyanidin-based anthocyanins, so cyanidin and peonidin, but not in malvidin. Here we have the same level in 2016, and here we have a higher content of malvidin in control than in SWE. This probably because control vines invested in more stable anthocyanin component as malvidin, to compensate the reduction in the anthocyanin biosynthesis. We deepen the analysis on anthocyanin, and we calculated the ratio of methoxylated to non-methoxylated anthocyanin and the percentage of methoxylated anthocyanin on the total. And also here we have in both here at full maturation a lower ratio of methoxylated to non-methoxylated anthocyanin and a lower percentage of methoxylated anthocyanins on the total in SWE than in control. We know that in stressful condition, hydroxylated anthocyanins were converted in methoxylated anthocyanins, so probably this lower ratio in SWE witness a major capacity to mitigate the stressful condition in SWE vines than in control. So in conclusion, this type of extract increased leaf gas exchanges, maintained a quantum yield efficiency of the photosystem 2 at a higher level, and has no effect on the water potential, lead to major grape balance with a higher content in anthocyanins and a lower content in sugar. And so these studies show that Ascophyllum nodosum treatments during the last stage of berries development may enhance the performances of Vitis vinifera, improving ecophysiological responses and quality traits with a major balance. And these studies support previous findings on Ascophyllum nodosum treatments on a plant acclimation to stressful environmental condition. Now I call on the Professor Mattii for other example of research in viticulture of our group.
Show me how it works.
These are some papers.
Okay. Good morning, everybody, and thanks, Marco, for inviting us to this meeting, because there is a very important occasion for us to present our research and to have new ideas from all of you, ladies and gentlemen, to have a new way to proceed in our research. I would like to give you a quick overview of research we are carrying out now in our department about viticulture. The first one I would like to talk about is a new project. It started just 1 year ago. It's a LIFE project. LIFE is a project that's supported by European Community, and it's carried out with several cooperation here in Tuscany. Zio Wine is a new product we are trying to test in vineyard to support organic matter lackness that we have in our vineyards. The partners, the coordinator is CNR, Institute of Terrestrial Ecosystems in Pisa, and the other partners are University of Florence, CMM, which is a winery, a biodynamic winery here in Pisa province, DN360, which is a company who sells zeolites, and Prima Forma, which take care of all the bureaucracy of the project, which is actually very complicated. The problem from we started is the organic matter. Organic matter is decreasing year after year in all our soils, especially in the specific cultivated soils. Vineyard agriculture soils in the 60s, they had 4–5% of organic matter. Now we are lucky when we have 1% of organic matter in our soils. That's because we don't have livestocks anymore. We have specific cultivations, so we don't have rotations in the cultivation. This is the main problem. Organic matter is one of the first problems we faced, but water stress is another one. I will talk later on climate change. I would like to see the climate change from another point of view, but actually we have several years in water stress. What happened really is that we have unusual behavior of meteorological conditions, unusual behavioral conditions year after year. If in the 60s we had 1 or 2 years every 10 unusual, now we have 6–7 years on 10 unusual. This year, for example, is quite unusual because of the temperature in June. Last year was unusual for all the rainfalls we had. 2017 was unusual for the drought we had in the year, and so on. Also, in the vineyards, we have a heavy metal accumulation, especially copper, which is used especially in organic and biodynamics vineyards. So this is another problem we face with this project. This project is financed with the total amount of budget is roughly €1.5 million, and is supported by the European Community with €860,000. The duration started, as I said, last year, and will end in 2022. The soil is, well, we heard this morning about soil. I don't have anything to add about soil, but we have a progressive degradation of our soils. Especially until the 80s, we didn't take into account the importance of soil, the importance of soil conservation, which is the base of our production. So especially the reduction in organic matter, with this reduction we can have a decrease in soil fertility, agronomical, but also a decrease in nutrient availability from the plants. So it's a lackness for everything. One of the main problems in vineyard of the lackness of organic matter is the erosion. We are cultivating our grapevines, especially in Tuscany, in steep slopes hills. So we have a problem of erosion that leads to a series of problems like soil compaction, like landslide, and so on. Moreover, the loss of organic matter in our soils can take to biodiversity loss. We have less, I would say, complicated population in the soil. That means that we can have more facilities for pathogens, because we have less balance, less equilibrium in the soils. We can have contamination also in our soil by the heavy metals that we said before. So, why to increase organic matter? Organic matter is important first for nutrition of the plants, because it's a long-term nutrition method. Can increase water retention capacity. Before we heard about soil availability water availability in the soil that depend very much from the presence of organic matter. Can have a buffer activity on the soil, so can make the pH more stable. Can control the cation exchange, so the availability of the nutrients of the cation. Can reduce pollution and improve the aggregation in the soil. That means the soil structure. Easier substrate for roots to grow. Which way to increase organic matter? We have the green manure since the Romans, maybe earlier, that is quite widespread in our viticulture. But there is another method, very useful, which is the compost. Now we are able to collect a lot of compost. We can compost it, so inside the winery, inside the vineyard. It is obtained for organic waste, organic byproducts. The effect of compost is on the microorganism in the soil. That means soil protection, that means benefits on the environments and effects on the plant itself. Those are some reference about the importance of organic matter in the soil, compost in the soil. Actually, we did a research on the use of compost here close to Florence. The use of compost in vineyards, in new vineyards and in an old vineyard. In old vineyard, we distribute the compost that comes from the residuals in the domestic use. Actually, we obtain interesting results in increasing the capacity of Sangiovese to accumulate anthocyanins. We had better anthocyanins content in the grapes after the subministration of compost in the vineyard, and better structure of the soil. And then we arrived to zeolite. Matching these two components, we made a new product. What is zeolite? Actually, the translation from the ancient Greek means the boiling stone. It's a stone, actually, grinded in different diameters that can be useful because it has some interesting properties. It can accumulate water when it's cold and release water when it's hot, which is perfect in our conditions. It can also accumulate heavy metals and trap these heavy metals in its net. Can also accumulate some elements like nitrogen, for example, and release slowly this element. We have, of course, long-term release nutrients, but they are very expensive. If I can use the zeolite, and for example with urea, which is the cheapest nutrition with nitrogen nutrition, would be very useful for our vineyards because we can give in 1 or 2 solutions and then release very slowly during the time. So matching these two, this is a treatment with zeolite in the vineyard, and we have some reference about the importance of zeolite. Matching these two characteristics, compost and zeolite, we made, the idea was to make a new product, the Zeo wine. what are the aims of the project? First, we want to improve agronomic and biological fertility in the soil, to have a soil more suitable for the life of the plants. To reduce the chemicals, because when we fertilize, especially with nitrogen, we have a lot of loss of nitrogen that goes in the water, and then we found in the sea eventually. So we can reduce this chemical because it's blocked by the zeolite and released slowly in the time. We can increase the sustainability of the winery because for the compost we use all the byproducts in the grape and wine chain. So we can make a sort of closed circle of these products. We should have more stability in the yields. We have a big fluctuation due to this climate, this different climatology in the different years. And we should have more quality in grapes and finally in the wine. So we want to produce the wine, the wine chain waste, byproducts and waste, they will composting with zeolite to make the zeo wine and return back to the vineyard to increase the soil characteristics, increase the sustainability, and to have better conditions in general. The aim is to have environmental benefits, economic benefits, because of course the economy must be to take into account because the final goal of the production is economic, of course. And social benefits, in one word, sustainability. So how can it face with climate change? Well, it's not directly related to the climate change, but we can reduce the emissions because we can use all the waste in the chain of production. So using all this, all this product, we can reduce the use of product coming from the outside of the chain. So we can decrease the carbon emission in the chain of production, which is actually very high. Another project, I hope to have time. Another project that we are starting is quite similar to the one I just talked about. It's uva preziosa, which means precious grapes. It's an exploitation of those byproducts in a different way. We know that in winemaking we have a lot of byproducts. I was trying to give some numbers. We have unripe grapes. What does it mean? In high-quality viticulture, we apply cluster thinning. Before maturation, when maturation is starting, we remove a part of the clusters to increase the quality of the remain. So these clusters are thrown away. We can use these clusters to extract, we will see in a while, to extract some products. We have marks, pomace. We have leaves, we have stalks, we have seeds. We have a lot of products that are usually thrown away. We have in Tuscany 370,000 tons of grapes per year, roughly. There is some variation, but more or less this is the number. We have, from those, we have 100,000 tons of pomace. We have 15,000 tons of leaves. We have 10,000 tons of stalks. How can we use these products? The objective of this research, that by the way is financed by Regione Toscana, is the valorization of those byproducts and of the waste, and utilize all the product. For example, with unripe grapes, we can make an extraction and to take away the antioxidant products, product with antioxidant activity. They can use with supplements for human nutrition or animals, in cosmetics, and as additives to come back to the chain, to the wine production. Pomace and lees, we can apply to them a new vinification technique. It's registered since a few years for quality wines, the extra velvet, that can be an extraction of, is actually a wine concentrated with polyphenols, especially polyphenols in red varieties. So the residual polyphenols, that they are a lot in the pomace, especially in the pomace, can be extracted and they can come back in the wine itself or make new wines. And from the seeds we can make an extraction with no solvents but with high pressure. It's a cold extraction to obtain oil. To obtain oil for cosmetics from diets itself. So it's a very light oil with low calories. What happened after? Because we have also a waste from this process, but with the waste, there will be composting for biogas production, quindi for energy, so for energy production. And then they will come back to the vineyard as a compost to close the chain. In the future, we can also match these two research projects and add zeolite also to this. Of course, the compost here is less, but we can match these two and add zeolite and make this new product. It's already started, this project. It's just started, so I don't have so far any results on this. We are just starting with the extractions. The last topic I would like to talk about is water stress. Last year, I was called to make a seminar in a school in Montalcino that we were talking about maturation of Sangiovese facing the climate change. So I tried to find some data about climate change, about temperature in Montalcino. I found this. This is the CIR from Tuscany. This is the website that collects several, I would say many meteorological stations spread throughout all Tuscany. We have a lot of data. I promised to myself to check this data also in different locations. But this is the data, temperature data in Montalcino from 1998 to 2018, last 20 years. This is the trend. It's just, I repeat, one station in one location, just one. I don't want to add any comment. This is the average of temperature from the 1st of April to 31st of October. It's not the annual, but it's the time in which the grapevine grows. Anyway, just for information, I'm carrying out a thesis with a student that will be graduated next week in Chianti, in which we have an increase in temperature, actually. There is an increase in the last, I guess, I don't remember if 30 or 20 years, but there is an increase, a slow increase in temperature. Actually, we have heat stress, for example. I remember 2012, Marco, help me, or 2011, I don't remember in which we have a strong heat stress starting from the middle of August. We had a beautiful season until the end of, the middle of August. Then we have a heat stress, temperature rise up of around about 10 degrees in 2 days, and we have a very strong heat effects especially on the early maturing cultivars. 11, not 12, okay. No problem, Federico, I trust you. So, climate change, we can have these kind of problems, burning in the clusters. But I was very impressed from the data of Dr. Cassardo. Very, very impressive, because it shows very clearly that this kind of variation due to climate change starting in the 80s. My question is, but before the 80s, there was the climate change, or the climate change starting in the 80s. But what actually started in the 80s? This one. Changed everything in the vineyards. In the 80s, we had a big renewal of all our vineyard plantation. We changed soil management. I made my degree thesis in '84 on ground cover in vineyard in Tuscany. It was something new. Nobody was doing ground cover in vineyards. We made canopy management. If you remember the canopies in the 60s, in the 70s, I mean the viticulture made from the European Community founding, FEOGA foundings, were completely different from the viticulture we have now. We have a different canopies, very low surface, very low leaf surface versus what we had in the 60s. Now we have a total leaf surface in a plant, in a vine, about 3 square meters. At the time, you had 7, 8, even 10 square meters of leaves. We change the varieties. At that time, in the 70s, until the 80s, we had quite a few of Cabernet and Merlot and Sauvignon and Chardonnay. We had in Tuscany, I mean, we had the Canaiolo, we had Colorino, we had autochthon varieties. Now we don't have those varieties. But mainly we changed rootstocks and planting density. All the viticulture of the 70s was on K5BB as a rootstock. Now, I think the percentage of KBB is insignificant. Now we have 10114, 1103, 110R, and so on. So we have everything different. Of course, we have different behavior. I don't want to say that it doesn't exist, the climate change, of course. Otherwise, Marco will kill me. But we have changed everything in the vineyard. And we say that all the responsibility of the climate change, are we sure? I don't want to give an answer to this, but I want to give a question. And please think about this question. Of course, all this change, all these parameters, all the change drive the viticulture to the same direction. They drive the viticulture to high alcohol wines, which is a problem now. I remember in the 60s, in the 70s, there is I couldn't say to the microphone, but I shouldn't say to the microphone, but there was a big amount of sugar used in the cellars. Now, the main problem is to have too much alcohol, not too low alcohol. Too much alcohol has negative effects on fermentation and testing. I have 10 minutes. And mainly, we have, we heard before, an imbalance between technical and aromatic maturation. Aromatic in this case because we are talking about Sauvignon Blanc, but I would say the same as imbalance between technical and phenolic maturation. If I want to wait for the good phenolic maturation, we didn't talk about phenolic maturation in the 60s and in the 70s. We didn't even know about phenolic maturation, but now we are concerned about phenolic maturation, and to wait phenolic maturation, we increase the alcohol content. What we did is to try to keep the vineyard, the grapes, not under stress. I mean, to make, we use the pre-down water potential to drive the irrigation. And what we established is to have some grapes, some vines, from 0 to 0.3 megapascal of our pre-dawn water potential, which is no stress at all. Then we establish another treatment from 0.3 to 0.7, also in pre-dawn water potential, and the control with non-irrigation. We found a good year, and actually what we found is that the water situation we obtained was actually what we would like to have, because this is the pre-dawn water potential in which only at the end we arrived to 0.4. So it was exactly what we had. I go quickly to some results of this. We have two different vineyards in which we can see a fluctuation in photosynthesis, but a separation of the photosynthesis, especially starting from this data when we started actually the irrigation. Before we didn't need any irrigation. So we have an increase in photosynthesis with irrigation, with better water relation in the vines. Especially we can see this in stomatal conductance, in which we can see that with no stress treatment, actually we didn't have any stress, because we arrive close to this threshold, which is the moderate stress. But in this case, we didn't reach any stress. A very interesting correlation we obtained between water potential and stomatal conductance, but 5 minutes, that will be enough. Thank you. But actually, it's the same you can find in the literature. This is the reverse. The two axes are reversed, but the meaning is actually the same. Very interesting was, especially in this vineyard, not just in this one, but in this vineyard was very important, the reduction in sugar accumulation. At the end, this is a white wine, so to have too high alcohol is not very good for the wine itself. But moreover, we had a better acidity, and all of you knows the importance of the acidity in the white wine. So we have a highest acidity. But mainly, we had those two graphs are very important, because we had an increase in the aromas, both in the cysteine and glutathione. So we had this increase. And also, we have a decrease in the cluster temperature. So this irrigation, this better condition as in water, allowed to have a lower temperature in the clusters, and probably this lower temperature plays an important role in the accumulation of the aromas. Very quick, I would like to show you something. Actually, we made it together. So in the canopy management, we set up a new system to study canopy management in vineyard. We build this system, those are balloons, to measure the whole canopy gas exchange in the grapes, in the plants. This system can measure the CO2 accumulation and the water emission, the water vapor emission in the whole canopy. It's a quite complicated method, but working, we obtained a system which is called plant analyzer for CO2. Those are some moments in which we were building the system. This is the final results that can control all the balloons and can accumulate the data. It's a sort of data logger. So we can have data at very low intervals, every 8 minutes, throughout days and days. We can set up the system in spring and remove at the end of the summer. So we can measure very interesting aspects. For example, this is something we measure. We had 2 different canopies, an horizontal trellis and the VSP, vertical shoot positioning, the normal training system we have in Tuscany. And you can see actually the two systems are quite different. The first one is irrigated, the second one is not irrigated. But anyway, we can measure that in one day we can, in one day we can have about 10 liters per plant in the horizontal system, and a half, roughly 4 or 5 liters of water transpired during the day. So with this system, we can set up, for example, the irrigation. We can understand how is the water consumption and set up eventually the irrigation. Just a quick overview. I don't want to ask you to read all these names, but we have in the University of Florence, starting from the 80s, we collect the autochthon varieties spread throughout all the Tuscany. And we collect in a germplasm repository, which is in a winery in the south of Tuscany. We have all these varieties. What the importance of these varieties? Probably all those varieties will be important because it's a source of DNA. So far, they are there, but they can use in the future to make breeding, to make cross, to face the climate change, to face the new wish of the viticulture world, to face new product we would like to spread on the market. So it's an important collection that we are taking care of it day after day. All this work, they won't be possible without all my beautiful and precious postgraduate students that are working, even today, I guess. They are working in the fields and taking data and elaborate. One of the samples is Linda, who talked a few minutes ago. Thank you very much for your attention.